This morning we are going to talk about this practical session in the laboratory of عسير Central Hospital. Here we are in the blood bank section, and we have our student from level 8 laboratory science student. This course is actually for quality control and laboratory management. And this laboratory management, we are going to take it as a first part, and then we'll talk about the quality control in this section. Regarding laboratory management, we have 13 points we are going to talk about. First, if you have been assigned to work in this place, and you have a letter, and then what shall you do? How you start? An idea? How you start your first day in this section? Any idea? No idea at all? It's okay. First of all, we are going to draw laboratory map, which is sketch what is the content of this place. And this sketch, I have some drawing like this, in which we are putting our shelf and tables and what is there, refrigerator, incubators, any similar picture, and in a drawing. Then we have to differentiate it in section A, B, C, for example. And then we put our higher shelf, H, and lower cupboards as low, or L. And then you can put also your office or table, and your hood or incubator or refrigerator, it is in similar places. So you have a map like that. Then what after that? We are doing numbering. We will put some numbers on these places, like this. So we have, for example, A1. That means the station or the office will be number 1, A1. We put office, for example, the content is office. And then we put AH1, which is section A, high shelf, number 1. So when you open this cupboard, you will find, for example, flask or beakers or whatever content is there. So others like AH2, and AL1, this is the lower cupboard. What is there, for example, gloves or plastics, and so on. So the B section will be similar, and C section will have the similar things. So you have a very nice view and sketch of your laboratory component. Then you put some remarks for checkup. And this should be mainly contain date of checkup and signature of the person who did the checkup. This is the way how you keep your things updated and followed. Okay, so this is clear, huh? So the second step we are doing is the equipments. طبعا لازم تخلي بالك للequipments. You keep your attention to your equipments, and they are valuable. Okay, and you need them to be updated and working fine. So equipments like what? An example of equipments in the lab? Shakers, refrigerators, okay, and incubators, and all these things. Huh? So regarding the equipments, you need to put two important things. Working steps, sticks, very small stick, how it works, okay, on and off and all the step of working. And then very nice small stick regarding the instruction for quality. So you need to clean it every day, for example. You need to change the water every week. You put some remarks or points for equipment, for quality control regarding this, for example, equipment. Then the reagents will be the third step, or the fourth step actually. Reagents can be liquid reagents or powder or any sort of reagents. So these reagents are very important. You put similar reagents together. For example, you have acids, put the acids together in one shelf or one cupboard. That's clear? And then you put your list of reagent in each shelf. For example, I have hydraulic acids, for example, lactic acids, any type of acids or alcohol. And then you put the expiry date, which is very important. For reagents, you have to keep in mind your expiry date. The fifth step regarding the management is the personnel. Personnel means people who are working. Okay, so you put a list of your personnel. For example, in this section, blood bank, you put a list. Number one, we have how many consultants, for example, how many specialists, how many lab technicians, and others, if you have workers or others. Okay, and this should be in the list. And then you can add the specialty of each, I mean certificates of each of these persons. Okay, and then you can add also the training period they have spent. For example, X have spent some training period in Riyadh, in مثلا hematology or something like that. So you can keep updated. Your personnel is trained every time and then. Okay, and then number 6, the operational manual, which is very important. Operational means working. So we divide the operation manual. You have to have a manual of operation, and this manual should be divided in 3 sections. Pre-analytical steps, مرحلة ما قبل الفحص, and intra-analytical and post-analytical. This is a pre-analytical. Mainly you are taking care of your samples. Okay, how you collect the sample, who is bringing the sample, okay, and how you prepare the sample before starting. For example, you need to do centrifugation and other sort of function. And then labeling, which is very important, labeling. You put the ID of the hospital file and the patient name and age and so on. And the analytical steps, which is intra-analytical steps, this is the working steps. Also, you put some remarks on the steps. For example, you have to do this, you add 15 ml of this, and you fix it on a slide, and like that. So someone will come and see, he can do it. He can follow a new one or new any paper, he can follow the steps. Now, the post-analytical steps, which is important regarding the reporting of analysis, I mean the results. So how you report your result? Is it a hard copy on a form, okay, or in software? You put it in your computer, so you keep it in your records for longer time, and you can retrieve your results. 7, which is important, the working environment. Environment means the temperature, the light, the ventilation, the spaces, the furniture. You need chairs, you need office, you need tables, like that. So this is what we call it, environmental environmental situation. Right? This is important regarding the laboratory management. For this course, we are dealing with this section of blood bank, and we have this form, and this form should be filled by the student, and you write your name and your ID, the date of inspection or being trained here in this section, and this is Asir Central Hospital, and this is Abha General Hospital, and the different section here is blood bank, serology, and microbiology. You tick on the suitable place, and then you list the activities. So let us start by listing the activities of this section. What activities is done in this place? What we are doing here? Blood donor, blood donation, yes, and okay, and then we have some tests before we do the blood donation. For example, we have first of all we take some history, and we have some sort of forms, and we fill in. This history should contain what is the last time you have donated, and have you any disease like diabetes, hypertension, you have any problem like, for example, hepatitis before, or malaria. This should be filled on the form. And after that, you do some clinical examination for this donor, and you can just weigh him, take his blood pressure, and take his weight, yes, weighing, and take his blood pressure and so on, and pulse and so on. And if he is suitable for donation, and you are going to do some tests, which is important for us. So these tests are actually summarized in this form. It's a very nice small card which should be done for our microbiology department. Number 1 is HBsAg. What is this? Hepatitis B antigen. What is the significance of this? What's the importance of this? If it is positive, what does that mean? That is hepatitis, so you will not take blood from hepatitis positive, صح? And then the second one, which is HCV virus, yes, and then HIV, which is immunovirus, and HTLV, which is a human T lymphocytes virus. And then we have, if you can see, this is 1, 2, 3, 4 are viral. These are viruses. Then we go below, we have VDRL. What is a VDRL? It is a test for syphilis, and the abbreviation is VDRL is Venereal Disease Research Laboratory. Venereal means sexual. Venereal Disease Research Laboratory. This is a test which is non-specific for syphilis. It is a screening test, and it's usually done as agglutination test, antigen antibody test. They are doing this test in the second place there, and we can see it later on. And if it is positive, you have to send this patient to do specific tests for syphilis. And we have different type of specific tests. Anyway, then we go to the MP. What is the MP? Malaria. So we have parasites, we have bacteria, we have viral. 4 types of viruses we have to screen for. And then we have the last one, which is ABO. What is ABO? Blood grouping. And I think we have some small tests for blood grouping, and this can be done here with kits, special kits for blood grouping. And these kits actually contain of 3 types of vials outside. So we can see it outside, blood grouping, yes. So we have this actual 3 types of bottle or vial. What is this one? What is the blue color? Anti-A. So what is this means if it is positive?
The person have antigen in the A, in the red blood cell.
So he have B, صح?
Yes.
So he's blood B group. So the other one is anti-B. The same, B is the reverse. And this one is
Anti-D.
the clear one, anti-D, which is rhesus. Yes, okay. So this is the 3 things. So what actually you do, you can put it in a slide or you put it in a piece of wood, and you put your drops of blood from the donor, 3 drops. And then you add 1 drop from each bottle in these 3 drops. At the end of the day, you will find clotting in certain place, and you can read it. Fine, very good. So we know they have universal donor and universal recipient. Okay, the O usually is donor, and while ABO, usually they are recipient. Recipient means they used to take from others, and they are not suitable to give. Okay, right? This is what is abbreviated here, RHD, R rhesus, which is, yes, antigen, which is positive or negative. Okay, and if you can see, this is the form I told you about, which should be filled first, containing some questions, 16 questions, for example, in Arabic, but we can translate it. Do you want to, have you been donated last weeks, for example, 8 weeks before? Because this is a shorter period. You should not give blood during 8 weeks before. So longer duration is better. And have you been donated before? Have your wife donated? And so on, different names, especially what we are interested in. Have you have hepatitis before? Have you been diagnosed as HIV? Have you leishmania? Have you any Rift Valley or any viral disease, Ebola, and so on? And then we have also to tick these diseases here, malaria, TB, leishmania, and many different names, and diabetes and cancers and so on. And then we have at the end to do our clinical evaluation by weighing and taking the CMC. So when you find some tests here, you can read it, and then you can see our sample, positive or negative. How? By which way? By using by eye, visual, direct vision. So you say the test is read by direct vision. Okay, and then what do you learn from this section? Should be learned here. Have you learned anything more than you have learned in the university, or similar? So anyway, at least we know how we are processing this in these places. Starting by filling this 2 questionnaire or 2 forms, and then we do our donation. And then you comment on this, is the place is good or not, And so we go to the other inside section where the processes are done for this test, most of them, and we can show you some tests there. Okay. And this is a place where the donors will stay, relax, comfortable, and you have to relax him before you take his blood. And this is some instrument for blood pressure and pulse and so on. And this can be detected before taking blood. So we go inside. And Assalamu alaikum. Assalamu alaikum.
دكتور هي بس البكتيريا الوحيدة اللي
MP, VDRL, venereal disease, research laboratory. Yes, this is for syphilis.
هي اللي تكون بس مؤثرة بالدم يعني.
For blood donor, it's from blood.
أنا أي بكتيريا ثانية تأثر.
No, other bacteria are not important يعني for donation. And they usually healed or يكون a patient all he's ill and you go and send him for treatment. لكن السيفليس لأنه stay long in your body. Sometimes حتى تكون asymptomatic, no symptoms. So you are carrier, which is serious. Okay, we see here this is a bag of blood. This is ready for donation, and this is written here A positive and the ID number of the donors, which should be used. So the question here, if you can have a look to this, we have refrigerators where we have, this is important, this is centrifuge for separation of serum because we need to take antibodies. This is your colleagues. Assalamu alaikum. He's doing the identification of the blood packets, which should be around 500 cc's, and it should be clear you know it is A or positive. Why? Because the problem of hypersensitivity, allergy. If you give someone a blood group which is not suitable, he might die, especially in night. But sometimes if you are emergency and you don't find any suitable blood group for your recipient patient, you give him anything. This is first of all. The hint, better if you have O negative, which can be donor for anyone, right? So if you have a look to the refrigerator, which is nice refrigerator, clear shelf, A plus, O plus, O plus, and B, and so on. This is actually not to make any mistakes. And it can be only on the bucket, like it also should be filled in this. So the question is that if you have frozen blood and you have fresh blood, which one you will give it to your patient? Which one is better for your patient? The frozen one or the fresh one? Thawing, frozen and thawing. And why? What's the reason? Is there any reason? So sometimes some people they say because we don't want to have it as expiry date and so on. So it is economical rather than scientific. But there is scientific background for this. Why you take the frozen one? Because the freezing itself is sterilization. It's killing other microorganisms. Freezing nafsu, it is a way of sterilization. Especially syphilis. VDR can be negative, but also can have some syphilis bacteria. Which is the bacteria name of syphilis? Treponema pallidum. Treponema pallidum, which is spiral bacteria. So the treponema is sometimes give you negative VDRL, and it's not that sensitive test. But if you did freezing for 2 days, 48 hours, you are going to kill any suspicious bacteria. So your patient, for him, it's better to have frozen blood than fresh blood, unless you have some certain things like platelets or other blood component. But if you want whole blood, it's better to give him frozen blood. Okay. So in this place also they're doing serology. مش كده تعمل serology هنا ELISA and that one. So ELISA is there and with the serology. And this is for syphilis or hepatitis. Okay. So we'll go to the section inside. You go with us? Okay. So we'll continue, معلش. This is a PCR section. And they have a very nice instrument. حوالي كم؟ 10 مليون ريال, 10 million riyals or something like that. Very expensive device. طبعا starting from maybe from here or from here. From this one, huh? You upload your samples here. For
bowling.
So okay, can we have some idea?
No, you can't.
No, no, you just give some idea, and then I will add. Okay.
هذا for bowling. This
Yeah.
for prepare the sample. This for amplification.
For identification. So you have 3 steps.
This step, yes.
Yes. So this is where you have what disease you are screening for? It's
For hepatitis B virus.
Hepatitis B,
virus,
C virus.
HIV 1, 2.
very nice. So you are doing the 4 viral section here. HBS.
This ELISA.
This is ELISA.
This for ELISA.
Mm.
PCR for hepatitis B virus, C virus, HIV only.
I see. So that is by serology. That's by serology, and this is by
Yes,
PCR.
This by PCR.
RNA, DNA.
DNA, RNA.
RNA. So this is a PCR section, which is very specific, very sensitive, and very rapid test. And there is software here where you can register your. So it's the real-time PCR.
Real time.
Real-time. There is no gel. No
real
gel.
time.
That means this is a real-time PCR. You'll get your result on the screen in shape of arrows and waves. Can we have any idea of?
Yes, you can.
Thank you very much. Just quickly. Thank you.
The computer is closed because
طيب will take some time. Anyway, we'll have waves. ها؟ أنت عندك حاجة طيب printed?
Yes.
Okay, yes, thank you very much.
This interpretation.
Yes. All right.
This, for example, Mikhail General Hospital.
أيوة.
The target non-reactive.
Yes.
Mean no virus in the sample.
Yes.
If reactive, maybe here no reactive or non-reactive. If reactive, give me reactive only.
We'll give you a
This
tita,
mean-
a number. We'll give you a number.
No, not give me a number. Give me only reactive.
Positive,
This,
negative.
This qualitative, not
This,
quantitative.
yes, very nice. Okay.
It's qualitative. If reactive, I don't know. Maybe hepatitis B, maybe hepatitis C.
I see.
Maybe by ELISA I can detect it.
I
Or
see.
send by
طيب quantitative ما عندهم? Why?
quantitative. الحين فتح يا دكتور في
في
مركز
الأس-
السموم.
I see. Because الميزة طبعا of the quantitative for the qualitative إنه will give you the viral load.
Yeah, viral load.
يديك الفيريميا
Quantity
how
of
much.
virus.
Yes. And this is like titer, like serology,
Yes.
like ELISA. So if you have a viral load, for example, 500 CD for HIV مثلا, so you think this is a serious condition for this HIV patient, like that. So the viral load is more specific for the situation of the patient. You can adjust his treatment or you can send him
also
home.
very specific and sensitive, doctor.
This one.
Yes.
But it is positive, negative.
Positive, negative.
So
only.
qualitative.
Only for screening, for
For
donor.
screening donors, yes. And if you find someone positive, what are you do? You send him to
If I,
serology?
Yes, serology.
Mm.
If positive also,
Yes,
send to Riyadh to
yes.
because
For-
now هذا I'm not open.
Not
Still,
doing yes, system. Okay, thank you very much. شكرا.
Welcome.
Anyways, this PCR actually is very expensive lab, like very useful and very specific. And you have to have some other equipments like centrifuge, rotators, and water pass, adjustable pipettes, specific things like. Do you have any other things specific for PCR lab?
No.
Uh,
I can use
because
by
you have
hand.
closed
No
system.
use
The
manual.
closed system.
No
Yes.
use the hand.
No manual.
Only
Everything
machine.
is
All
automatically.
else, yes.
Yes.
Separate
This is
by
the different. Yes. This is why it is expensive, and the contamination is a problem.
No contamination.
No contamination, because if you have contamination,
Also
you get
transfer from ampli to Tacman by automated. You see.
automated from it goes through like that. Very nice. So there's no way for contamination. So you'll have your sample positive. That means 100% positive. So you'll not be false positive or false negative. Huh?
How many samples?
Usually, I think we take.
I have 6 rack.
Mhm. Yes.
This 3 batch, 6 rack. Okay. 126 sample.
Mhm. Too much,
Maybe-
mashallah.
Yes, too much.
Mm.
Maybe take from 9 to 10 hours to
All the results. Very nice, والله. Thank you very much. شكرا جزيلا. بارك الله فيك. الله يسلمك. Here, this is the serology section.
In this section, we are doing screening for antibody for any infectious disease, autoimmune disease, and also we are screening the immune status of patient. Most of our tests are doing by ELISA technique, and for autoimmune disease like ANA, anti-DNA, we are testing this test by immunofluorescence assay. Other techniques are doing by ELISA also. Most of our tests are manual, but some for ELISA tests we are doing by semi-manual technique. Other tests, we are also screening for IgE total and IgE specific. Besides that, we are also following HIV patients. Among these tests doing by this, for this patient, we are doing screening HIV. Any test positive, we are doing confirmatory test. And also we are doing some marker like CD4, CD8. Also, we are doing other tests as agglutination tests, most of the tests for Vidal test, Bruxelles test, rheumatoid factor, and CRP. Also, for parasitic disease, we are screening by hemagglutination technique, like amoebiasis, echinococcus, leishmania, bilharzia. And also, we are using immunofluorescence assay for principally for ANA, anti-DNA. That's all.
Yes, thank you. شكرا دكتورة. جزاك الله خير. Thank you. Okay. So, what samples we use here? We use actually blood samples, and we are fond of having serum, not the whole blood, because we are detecting antigen antibody. So we are interested in the antibodies sample. Usually there is a form where you have all the tests or the activities run by this section. Sometimes some of these tests will not be available, maybe problem of reagents or something. Anyway, most of them, which is important one is hepatitis and HIV, because these are serious diseases and we need to confirm the diagnosis by serology, which is so when you enter in serology lab, just remember, we have quantity and quality. Okay, Arabic means كمي and نوعي. So the نوعي, which is quality, usually it is positive, negative, and actually mainly it could be like agglutination test. You put drop on a slide and just you see it is positive or negative, like that. Okay, like pregnancy test, like Lancet's test for streptococcus, like that. So it will be positive, negative, and the serology for quantitative, which is numbers, will give you titer. So you have to have something called cutoff point, which is a point by which you can determine your positive sample and negative sample. And this can be just imaginary. You can calculate all the measurements and you took the average. And average اللي هو المتوسط. And you say above this average is positive, below is negative, like that. But all this actually the company will tell you, because they will send you also positive and negative control. You have to run control with these samples. So the positive and negative control with your samples, they will give you also the cutoff point. They tell you titer of 1 over, for example, 100 or 128, for example, for mycoplasma, is positive, like that. Okay? So we are doing actually serial dilution. You know serial dilution? Yes. And then you can determine your cutoff point. So if you have a plate, and actually we have 96 plates, well, this is called well. These plates, each well is, or each arrow may be for one patient, because you are doing dilution. Huh? And then you can see the positive or negative by change of color. You can see some of these blue in color or yellow in color, and you say this is positive, for example. Or you can see the precipitation down. If there is precipitation down, it is, for example, negative sample. If there is homogeneous and no clotting down, it is positive, like that. So you can determine by eyes, and you can determine by reading, which is the reader, we call it. A reader will tell you how much this is, titer is, okay? So first of all, you have blood samples. You have to prepare your sample by separating the serum, and you take the serum for your, actually most of your tests, because there's antibodies in that serum, okay? And then we can apply it on your plates. Make sure you are not mistake or confused with other samples. Arrow number 1 is for patient name, flan, okay? Muhammad, for example, or something. So it is very, very important. And the plate is very small. Cannot give you space to write names and so on. you can have a draw. يمكن ترسم رسم and draw and you say plate number 1 is for this arrow for Muhammad, for example, and this one, and then you see. So we have to add positive and negative control. Why? If the negative become positive in your analysis or run, that means you have contamination, you have problem for your reagents, other things. So you have to repeat it, okay? And your positive sample, if it has become negative, that means also there is a problem for reagents, for example, or it doesn't work, okay? So ELISA machine is very useful, okay? And it takes the whole titer and give you the run. So actually we are doing dilution, serial dilution, and we are applying our antigen, and then we can see the reaction by doing adding certain stain or colors, and then we can stop the reaction, and we can do some washing and all this. So the very nice setup of ELISA should be full automatic. That means you have a washer, ELISA machine, and you have a reader. So you have 3 components. And actually the reader should be computer by printing your results. But some people, they're doing the washing and these things manual by pipetting this buffer, and then you clear it or dry it by air, or just knocking back on the desk, and then you retain and you add your other step of reaction. Okay, so ELISA machine, it has certain quality control measures. Okay, because you are depending on it for positive and negative, which for serious disease like hepatitis and HIV and so on. So you have to do quality control for ELISA machine itself. Okay, and usually the good quality control for ELISA is by doing the positive and negative control from the companies. Some people, they have known that certain companies are good, have good quality reagents. Okay, we don't want to make any propaganda for any company. Like you will, by work, you know which best company for reagents. Also, other things you need it, as I said, certification, because you need the serum. See, okay, that's the first step for preparation of your sample. Second, you have to do labeling. Okay, on this form, you give the number, ID number, or file number, or hospital number, and then you do tick on your which test you need to do. So give me some example for other not viral serological tests, non-viral. Any other, bacterial, for example. What sort of bacterial serological test can be done? We talk about one of us in the blood bank, which one? Syphilis, VDRL. Okay. So specific tests for syphilis and non-specific tests. Syphilis can be done by serology. Other things like typhoid, which is very important. Vidal test. Don't forget this name, huh? Vidal test, and brucella also. You know brucella? Brucella is humma al-maltiya. It's coming from? Why it is common here or not? Is it common? Why? Any idea? حليب العباير. Camel milk. Huh? So it's common here. مش كده يا دكتورة? You are doing this brucella, typhoid and
brucella and Vidal test as a stat test.
a screening test.
No, stat test. C'est-à-dire we are doing an emergency test.
As emergency.
And we are releasing the test within 1 hour.
1 hour.
1 hour. Vidal test. Vidal test.
But you wait for having quantity of samples, or one sample can be
for
run?
other tests, as we call routine tests, we are gathering the sample.
Yes.
And after we do our routine test. But for brucella, Vidal test, CRP, rheumatoid factor, we are considered as stat test, and we are doing within 1 hour.
Yes.
Within an hour.
So that is emergency. That will be more cost. And you are using one blade for one sample, and
Yes, for other tests, we are gathering
yes,
the
it will
sample.
be expensive. And
Yes, for
yes.
other tests.
Okay, يعني انت instead of waiting مثلا 100 samples to run them in one blade, for example, you can, for emergency, as Victoria said, you can put one sample an emergency in one blade. وبعدين شو البليد ده might cost you 10,000 riyal, for example. So you are doing one test for 10,000 riyal. Okay, so that is emergency. The others, actually, you need to wait. You keep your samples in the refrigerator, on a deep freezer, till you have a bulk of samples, maybe 30 samples, and you run them with each other, and it will be cost effective. So it will be useful for a patient. Okay, so that is a ELISA, that is a washer. See that bottles, and that is a ELISA readers, and can be run by computer, and you have your results computerized. And this is, for example, this is a printer. Also can be used as ELISA reader. We call it ELISA reader. And the reading actually by using optic density.
Yes.
And yes, and just optic density, and then you can see this is positive or negative according to the optic density provided by the company. طبعا الشركة which give you the test will tell you which optic density should be used for screening these samples. Clear يا شباب? Any question? No? No more questions? متأكدين؟ Sure? خلاص, just to fill this form, and we will see you inshallah in the lecture. Right, here طبعا هندركن طبعا. This is the back tick. Okay? This is a very nice time to see. This is the back tick. And what are you doing here, معلش briefly يعني? And tell them about this also. What is this? كلمنا عنها. What are you doing? Yes, tell us.
بس ندخل العينات blood culture في
This all blood culture? All these samples are blood culture, huh?
yes.
Okay. And this can be read by this instrument, laser,
Yes.
and then it give you positive or negative?
Positive.
And negative, okay?
Positive and negative.
Yes, this is a backtick actually, all right. And well, this can be done manual, but backtick will give you automatic readings for culture, okay? But we need to use this special sort of vials. Certain vials for anaerobic growth and others for aerobic. It will be clear from the color, or it will be written on the bottle itself, it's aerobic or anaerobic. And then the reading will be here. And this comes here to the reader on software, and you can have, yes.
patient name الاكسسن ليبلينج في اللاب يعني الرقم حقها نسويها. ديت كولكشن تايم يكون دايركت موجود فيها. الهوسبيتال سيرفيس ديبارتمنت في الام سي سي او او الاي دي. بعدين بس بالليزر تحط الجهاز و
After how long you will read it? 48 hours or 72 hours?
70 hours.
72 hours.
Maybe, yes.
Okay. And then what about the antibiotic resistant? It also comes here? Is there resistant for antibiotic?
Antibiotics.
Yes.
No, for culture, subculture.
For antibiotics, it
yes.
will not be here.
If there is positive,
Yes.
we will do a subculture and then
We put it here also
Yes.
again, and we will give you a reading here for
give
antibiotics.
reading. Yes.
I see. This is the only reading. Okay, thanks very much, shukran. So actually this is bacteriology section. We have microbiology, and we have pure bacterial section. And if you start from bacterial section here, there we'll collect the samples. And usually samples can be urine, swabs, sputum, can be blood for culture. All this can be sent for culture. And we use culture plates, commonly used nutrient agar and blood agar, and other McConkey agar for gram negative. You know this from reading previous studies. And you can do gram stain for positive and negative gram stain, and can do other stains, for example, GIMSA or silver stain or others. And you can do antibiotic sensitivity test by Kirby power disc, which is a sort of filter paper. You add it on the plates, culture plate, and you keep it for 48 hours, and you can see the positive sample, I mean the resistant one, will have clear zone around, or there is no clear zone around. So by measuring the zones surrounding the antibiotic circle, clear, Sheba? And you can say this is positive or negative. Alas, for example, you have tetracycline, and you can see the gross, is it there is clear zone around this, so that means sensitive to tetracycline. If there is no clear zone, you can say this is resistant to tetracycline, for example. Huh? So this is how you read antibiotic sensitivity test. And usually we use filter paper for common usually used antibiotic like penicillin, ciprofloxacin, or any common antibiotics. Then دكتورة فاتن is in charge طبعا. We are welcoming you, دكتورة فاتن, in this bacteriology section. Still is for the activities.
No, this is the processing area for culture. This is our processing area for culture. English or okay. This is our bacteriology media. Okay. This is how to receive your sample and to correlate data with the request. Yes.
The green one, what's it?
This is Khalid media. Okay. This is blood agar. This maconky agar. This is chocolate.
Chocolate.
Chocolate agar.
What? Clit. What's the clit? What is it? Clit agar يا شباب, green color. What is it? Why you use it for what? Yeah, tell them.
We use this cleat for urine culture. Okay. This is the main indication. Then after plating, you will plating should be in this safety cabinet. Okay. Like we do here.
After we do the ID identification.
No, this is just here, just receiving and the correlation of the sample and the making number. Here for plating, like this. Okay? Yeah. Then after he finish, he will incubate all the plate in these incubators. Okay? Yeah. On the second day, we take this plate for identification on this bench.
Microscopy or just by vision?
Yes, should be microscopy. Just
Can I have some example?
here you will correlate. This is you will correlate this. This is a plate. Okay, correlate all the plate because every sample has about 3 or 2 plate. Then you will see you write your professional diagnosis about this organism. If you need on bench test, you can do like catalase, oxidase, whatever. Yeah. Then after this identification of, or you can make gram stain also.
What is this? Yeah, professional diagnosis.
Professional, primary, you can tell this is gram negative bacilli.
Why?
Okay.
Why is gram negative? Huh? Any idea? Alone, color.
And also yeah, from the color, and it grow on maconky. Okay.
والله كان في يرسل شجلة.
After this item, you will start antibiotic sensitivity. Okay, on this bench. This antibiotic sensitivity. Like this, you process on molar Hinton agar, yeah, like this, and you will incubate. On the second, means the third day from you receive the specimen, you will start reading the sensitivity.
The clear zone is sensitive.
Also here we use for identification and sensitivity, we have 2 machines here, okay, for processing. And not all sample we process manual.
About the API.
Yes.
This one. So what is this one? API. What does it mean? API. Abbreviation. Analytical profile index. Analytical profile index. اللي في حوالي maybe 20 chemicals reaction.
If you will process your sensitivity manual, so you have to do this identification manual to know the type of the organism by this API system, which contain biochemical reaction. Okay.
You know how to read it? مش كده؟ By manual book or you can You just put some degree according to the color of the and you
Yeah. You will compare your color here with this. You have here this colored chart, as you see. And you write your data as positive, negative for every biochemical reaction. After that, you will collect a number, and you will translate this number from book. Yeah.
You get the right diagnosis. Okay. Now, tell us about the aerobic jar. Anaerobic jar.
We have here this is anaerobic jar, okay?
You will put
You will put it.
This carbon dioxide releasing kits.
Yeah, you need to put here HADA anaerobic container system like this. You will first you put your plate inside, then you will open this and put inside, but you must put indicator. We have here strip indicator to show that the media inside is anaerobic. It will show color change, yeah, as a control method for correct process.
Yeah, and then you put the jar in the incubator.
And the incubator, yeah.
Okay. What's anaerobic? Obligatory anaerobic bacteria, you know? Any idea? Anaerobic. أهم عائلة. The important family of Clostridium. كل التيتانوس Clostridium family. Clostridium tetani, Wiltshire, all these are anaerobic. This is gram positive bacilli. While the gram negative is anaerobic obligatory like macroids, groups, use bacteria and Bronchella. These are gram negative. Actually, if you know your patient clinical station like abscess, for example, any abscess can be gram negative, anaerobic, can be. Okay? So you need to use this plates. Any idea how many person you test? You have lancets, grooving.
Yeah.
This is the kits. What is the lancet grooving? Have you seen it before? Lancet grooving. This is the kit.
We have for this latex agglutination.
Have different groups, A, B, C, D, E, like that.
Yeah.
Local infection, you use lancet by adding it on the special filter. This one, موجود فيها grooving. This one. This put your drops, this, and then you add your culture swab. For example, you take a piece of the culture and you add it on these drops. So you find agglutination. That means this positive. Okay. And this is will tell you this is group A or B or C. Further. طبعا streptococcus usually عندها a lot of problems يعني. So we have to need to do so further test. You need to know it is lancet groups. You need to know hemolytic hemolysis. You know hemolysis? What type of hemolysis we have? Alpha, beta, gamma. Okay, don't forget this. Streptococcus عندها more tests you have to do. Okay, you know how to differentiate between strepto and staph? Staphylococcus. They are both cocci, they are both gram positive. So by vision you cannot, again you have to do some tests. Catalase and then coagulase to know it is staph aureus or not. Catalase positive it is staph, catalase negative it is streptococcus. Okay, it's very nice. Any question? Unless you fill this, you give it to me in the lecture, fill this form. The other section, you need another section and you can see it in the video. Okay, any other section, you can do blood bank or serology as you like. It's up to you. I'll see you in the university. Okay, Assalamu alaikum.
بسم الله الرحمن الرحيم والصلاة والسلام على رسول الله. السلام عليكم ورحمة الله وبركاته. I would like to welcome the King Khalid University, the medical field, especially laboratory field, for their coming and visiting in our Asir Central Hospital laboratory. My name is Yasser Abdullah Rai. I am lab technologist. I am also the responsible for the internship in the laboratory department. Laboratory department is our laboratory is one of the central and also reference department in the Asir region. In our laboratory department, we have 7 subclass of departments. 6 subclass of departments, which are blood bank and hematology, clinical chemistry, microbiology, histopathology, and these departments are the mainly group. Also, in addition, we have some of unique department like the PCR department, which is called NAT department, which is nucleic acid test, that is done for the screening test for HIV and hepatitis, whether it is P or C, by using the technique of the PCR. Also, we have one of the unique departments, we have the flow cytometry department that is used for the diagnosis for the some of leukemic and also the cancers that is related to the blood flotation. In Asir Central Department, for Laboratory Department, also we are concerning about the intern, that we sure that all the intern in this in the laboratory, taking all the information that they needed. Also, at the same time, they have taken the well practice, okay, and also that to be ensured that the intern has take all what it does he need for his internship, because when he inshallah will be graduated, can work in any hospital with no problems. Finally, I'd like to thank you, and inshallah we have more cooperation and cooperative work with King Khalid University. Wassalamu alaikum wa rahmatullahi wa barakatuh.